Hello everyone. Marker systems are tools for studying the transfer of genes into an experimental organism. In gene transfer studies, a foreign gene called a transgene is introduced into an organism in a process called transformation. A common problem for researchers is to determine quickly and easily if the target cells of the organism have actually taken up the transgene. A marker allows the researcher to determine whether the transgene has been transferred, where it is located, and when it is expressed. Marker systems exist in two broad categories: selectable markers and screenable markers. In this module, we shall study some of the features and applications of these two types of markers. The learning objectives are features of selectable markers, features of screenable markers, and applications of the screenable markers. Selectable markers are typically genes for antibiotic resistance, which give the transformed organism, usually a single cell, the ability to live and grow in the presence of an antibiotic. Screenable markers, also called reporter genes or scorable markers, typically cause a color change or other visible change in the tissue of the transformed organism. This allows the investigator to quickly screen a large group of cells for the ones that have been transformed. A selectable marker is a dominant gene which encodes a product that allows transformed cell to survive and grow under conditions that kill or restrict the growth of non-transformed cells. Selectable markers used in selecting transformed plants are those that confer resistance to antibiotics like neomycin, kanamycin, bleomycin, and hygromycin, or to herbicides like bilophos, glyphosate, chlorsulfuron, or naphthalene acetamide. Some of the commonly used selection markers include neomycin phosphotransferase 2, which is a bacterial enzyme that confers resistance to the antibiotic kanamycin. The NPT2 gene provides resistance by phosphorylating kanamycin, thus making it inactive. Hygromycin phosphotransferase 2 codes for hygromycin phosphotransferase HPT, which detoxifies the aminocyclitol antibiotic hygromycin B. EPSP synthase is an enzyme of the shikimic acid pathway that plays a role in the synthesis of aromatic amino acids. The enzyme that is genetically modified does not bind to the herbicide glyphosate that goes by the trade name Roundup. Transformed plants produces a functional enzyme and grow normally when sprayed with glyphosate, making them glyphosate resistant. The non-transformed plants die when sprayed with the herbicide, since they cannot synthesize the aromatic amino acids. The gene constructs made for expression of the selectable markers in prokaryotic organisms like E. coli or Agrobacterium include the marker gene with a prokaryotic promoter, example LAG-Z promoter, and a prokaryotic terminator like LAG-Z terminator. For expression in plants, the selectable marker gene expression is driven by an eukaryotic promoter like 35S cauliflower mosaic virus promoter or the nopalene synthase or NOS promoter. The marker gene is flanked by polyadenylation signals of eukaryotic genes like NOS. Screenable marker systems employ a gene whose protein product is easily detectable in the cell, either because it produces a visible pigment or because it fluoresces under appropriate conditions. In bacteria, the LAG-Z marker is used for reporting the successful insertion of gene of interest in bacterial plasmids due to disruption of LAG-Z gene expression. In plants, the screenable markers are inserted along with the gene of interest and report successful plant transformation. They provide a powerful tool for identifying transformed cells before the gene of interest is expressed. They can also identify the tissue that have and have not been transformed in the plant. Like selectable markers, expression of screenable markers is driven by the use of appropriate promoters and terminators. Selection of bacterial transformants for a gene of interest is carried out by using the blue-white screening technique. The LAGZ marker consists of the LAGZ gene into which a multiple cloning site is inserted. When the gene of interest is inserted into the multiple cloning site, LAGZ gene is not expressed. Hence, this marker is used for reporting successful insertion of a gene of interest in bacterial plasmids due to disruption of LAGZ gene expression. Colonies in which the gene of interest is not inserted appear blue due to cleavage of the galactose from the chromogenic substituted indole 5 bromo 4 chloro 3 indolyl beta D galactopyranoside or XGAL. Insertion of the gene of interest gives white colonies due to lack of LAGZ transcription. Since LAGZ gene is also present on the bacterial chromosome, it is important that the strain of E. coli in which the plasmid bearing LAGZ gene is inserted should not express the native LAGZ gene. This is done by the technique of alpha complementation. Beta galactosidase is a tetramer and each monomer is made up of two parts, LAGZ alpha and LAGZ omega. The E. coli strain used has the alpha fragment deleted from the chromosome, but the omega fragment is intact. In the plasmid, the LAGZ gene codes for LAGZ alpha but not LAGZ omega. Hence, beta galactosidase is inactive. Enzyme functionality can be restored in trans by the plasmid LAGZ alpha complementing the chromosomal LAGZ omega. Hence, then name alpha complementation. In plants, three types of screenable markers are commonly used. The first one is the luciferase genes from firefly, which is coded by the Lux gene. The enzyme converts chemical energy stored in ATP to light energy in the presence of its substrate luciferin. Expression of luciferase is detected by incubating tissue with luciferin and then placing it in contact with photographic film. The second marker is green fluorescent protein of GFP. It is a fluorescent protein from the jellyfish Aquaria victoria. In jellyfish, a protein aquarin emits blue fluorescence when activated. This provides excitation energy to GFP, which emits green fluorescence. Amino acids serine, tyrosine, glycine at position 65 to 67 in the protein spontaneously form the chromophore. Synthesis of the GFP fluorophore occurs spontaneously without cofactors. Hence, can be used as a reporter gene in wide range of organisms. GFP is small, tolerates both N and C terminal fusions with other proteins and can be targeted to specific cellular locations. GFP mutants and homologs exhibit fluorescent emission maxima ranging from blue to red. The GUS gene encodes the beta glucouronidase enzyme from E. coli and it has been extensively used as a reporter gene in transgenic plants. Since the enzyme does not exist in plants, its activity in transgenic plants reports successful transformation with the gene of interest. The main advantage of the GUS reporter gene system is the stable expression of GUS enzyme in plant cells. GUS activity can readily be detected by blue colored complex formation due to GUS activity on the substrate 5-bromo 4-chloro 3-indolyl glucouronate X-gluc. Fluorogenic substrates can also be used, for example, 4-methyl umbelliferyl beta D glucouronate, which has an excitation wavelength of 365 nanometer and emits fluorescence of wavelength 445 nanometer. GUS staining cannot be carried out in living tissues and requires fixation of tissues. The screenable markers have several applications. They can be used to confirm whether successful transformation has taken place when plants are transformed with a tDNA based vector carrying a reporter gene. The reporter gene's expression is independent of the gene of interest expression, which is an advantage when the gene of interest is only expressed under specific conditions or in tissues that are difficult to access. The screenable markers are also used to study gene expression. For doing this, a reporter gene is placed in tandem with the gene of interest and both genes are expressed by a single promoter. Expression of the reporter gene gives an indication that the gene of interest is also expressed. This works only if the reporter gene product does not interfere with the activity of the gene of interest and if protein folding occurs correctly. Reporter genes are also used to study the spatial or temporal expression of a gene that is driven by a particular promoter. The reporter gene is inserted adjacent to a particular promoter and expression of the reporter gene indicates the time and place where the promoter is induced to drive gene expression. The strength of the promoter can be estimated by comparing the expression of reporter gene expressed by the specific promoter to that of a reporter gene expressed by a constitutively expressed consensus promoter known to induce strong gene expression. Promoter trapping is another technique that uses reporter gene expression to identify promoters. The promoterless reporter gene is allowed to randomly insert into the plant genome. If it inserts adjacent to a promoter, the expression of reporter gene is used to understand where and when the promoter is active. All these applications have led to the widespread use of screenable markers in biology. Let us summarize what we have studied in this module. Selectable and screenable markers are important tools in genetic engineering. Selectable markers enable selection of transformed bacterial cells or plant cells and tissues from the non-transformed ones. They are usually genes that confer antibiotic resistance to the transformed cells or tissues. Let us summarize what we have studied in this module, selectable and screenable markers. Selectable and screenable markers are important tools in genetic engineering. Selectable markers enable selection of transformed bacterial cells or plant cells and tissues from non-transformed ones. They are usually genes that confer antibiotic resistance to the transformed cells or tissues. Screenable markers not only report on successful transformation, but also tell us whether the transformation is stable, whether the gene of interest is expressed, and also about specificity of expression of a promoter. These markers have greatly improved our understanding of the temporal and spatial expression of genes. Thank you.